many of them since, you know.
Is this actually a hybrid event or is it just for recording purposes?
Mostly for recording.
Okay,
Uh,
I was just curious.
yeah, the only person on Zoom is Vivian,
Okay,
but I did open it, like, if people ask, but I tried to. get
okay,
as
yeah,
many
okay,
as possible.
gotcha.
We'll see. if anybody's out
There's
there.
still a few people I
Okay,
can
cool.
see
You can see them.
in the line, yeah.
Zach didn't come?
What's that?
Zach didn't come?
No, we got, the timing didn't work out with in the lab, yeah, so it's fine. He's seen this many times.
Yeah.
Or I shouldn't say many times, but multiple times at least.
What are you guys physically located?
You know, do you know where like the I think it's biochemistry, the teaching lab is, like Harold Smith's old lab? Like, like if you're coming from, you know, where like Josh's lab is, that hallway. And then
Okay,
there's that like string of doors to go through, including like the vivarium access, and then you come out on the other side on the third floor, yeah.
okay.
Then that's where we are.
Yeah. Dear child, dear children. Get that right.
Someone just went around the corner.
All right, we'll make it 30 seconds to get the time to play. Hi, everyone. We have our Metabolomics seminar for our Lunch and Learn series today. Brad Smith is going to be presenting, and yeah, I'll let him take it away.
Thank you. Okay, so for those that don't know me, my background is in cancer metabolism, working previously in Hucky Land's lab. And together with Josh Munger, who some of you may know, and Zach Moore, who joined us over this past summer, we've been putting together and developing a new metabolomics focused shared resource aim to support and enhance the various metabolism related work going on here at U of R. And so I just wanted to take this opportunity in front of this audience to preview the new facility, what we have so far. And so I'll start by first just really briefly touching on metabolomics in the context of cancer, and then moving on to a general overview of our capabilities and planned services, and what should be maybe like 25-minute presentation. So then plenty of time for questions or any feedback at the end. But then also feel free to interrupt at any point during the presentation with any questions. We can keep this pretty informal. All right. So I think as many of us probably here know, changes in metabolism underlie many different disease states, and that this includes cancer, where among the numerous changes that are associated with malignant transformation and maintaining the malignant state, and critical to it are this is altered metabolic state. And so a metabolomics approach to probe this altered metabolic state is to essentially just measure and compare metabolite levels across different samples of interest as a way to potentially get at biologically and clinically interesting questions and tackle interesting areas of research. And this includes, for example, the discovery of metabolic dependencies or vulnerabilities that you could potentially then target for therapeutic intervention, in particular coming from material of advanced disease where we know the least about this. And then as another example, and relatedly, probing therapy resistance, where we can assess metabolic responses to a drug treatment to get a better understanding of underlying resistance and compensatory mechanisms that ideally we can then use to inform combinatorial strategies to counteract these responses. And so the ability to do the ability to do a more global or what is called untargeted metabolite profiling, where essentially we're trying to measure anything detectable or any metabolite detectable in a given sample, and which is the ability we're trying to bring here to U of R, allows for a more unbiased discovery of altered metabolic activities across samples of interest, including things that we may never even have thought to assess if we were just, as often the case, picking and choosing certain metabolites or metabolic pathways to target for measurement. And so this brings us into the instrument that the new facility is centered on, which is an HPLC coupled high resolution mass spectrometer with a couple of its key features that underlie its capabilities, being that it combines a high resolution and mass accuracy with the ability to do fast enough what are called scan rates, where it can acquire for most applications a relevant full mass spectrum multiple times per second as a given sample, and its components are eluted off the HPLC column over typically a 20-minute time period, and then enter the mass spec for detection. And so the main takeaway really to go away from this is just that it is this combination of accuracy of measurement and the speed at which it can make these measurements across a large mass range that allow it to really gear it towards this so-called untargeted metabolite profiling, where in a given complex biological sample, we can acquire the data needed to determine abundances and aid in the identification of the up to thousands of different compounds or metabolites that may exist within that sample. Okay, so then next, moving into I guess the three basic phases of what we do or how we go from a sample to data. we start with in what would be the pre data acquisition or sample prep phase, where we have protocols and equipment to, when necessary, homogenize and extract different metabolic populations as needed from diverse source material. And so this includes solid tissues, biofluids, cell culture material, including media analysis, et cetera. And then with these extracts, then we then also have a refrigerated auto sampler, which can hold all sample vials necessary for a given instrument run. And then one by one, these samples can be injected into the system, where they travel across the column while a gradient of different chemical mobile phases flows through the column well, and basically elutes different components of each sample off the column and into the mass spec over what is typically a 20-minute period. And then as these different chemical fractions of your original sample are ionized and then detected, and different ion charges and mass values are recorded along with the column retention time. So how long did it take that to come off? And then all these specific characteristics of what would be called features. So any ion that existed that is coupled to a specific time elute that it takes it to elute from, or the time it retained on the column, are then collected in this big, pile of raw data. And then we have the software to then take this big population of features, so different ion mass charge values coupled to a column retention time, and oftentimes specific what are called fragmentation patterns. And then we can basically deconvolute this and assign and generate abundance values for them, as well as assign as many compound identifications to them as possible. Okay, and then I'll just go into a little more now, like detail of each of these phases. So starting with this first pre-measurement phase, excuse me, I'm getting over a little throat issue here. Most projects, especially for new users, will typically start with some sort of consultation meeting where we'll discuss the project, what the needs are, what we can do best fits with whatever your question of interest is, your material limits, etc. And then determine sort of what the first needs are in terms of preparing samples for measurement. And so this can fall into two general categories, where we can do the sample prep, where we have the equipment to homogenize hard tissue or frozen tissues if needed, extract, evaporate these extracts, and then concentrate them in a smaller volume to optimize detection on the mass spec. And so we can do all this, or depending on the user's needs and abilities and access to their own equipment, they may do this themselves, where we can still provide detailed protocols and guidance on best practices for handling different steps along the way, but then they can also be the ones that actually prepare themselves if that's what they want to do, and have the capability to do so. And then moving on to the data acquisition phase, this essentially comes in two general flavors. One, and that I've been touching on so far, is for more discovery or hypothesis generating projects. And so this is our untargeted or global profiler acquisitions, where we're basically, there's what we plan to offer two different options, one geared towards profiling the hydrophilic compound world, one the hydrophobic world. And so depending on which of these, or maybe both, you want after, they involve basically measuring each sample with two specific different but complementary LC-MS detection methods, just to aim at providing as general and as comprehensive a profiling as possible of a different type of extract. Again, it's generally broken down to either hydrophilic or hydrophobic. And then, although I've sort of been focused on this more untargeted or global profiling, we also can support more hypothesis-driven projects where you're typically doing a more targeted acquisition And this can involve one of our previously developed methods, depending on what you're going after, or the development of a custom method that isn't something we've done before. And so this will typically involve just a single detection method per sample, per specific compound or group of compounds that you're really trying to optimize measurements for. And then sort of the fee structure is set up then for each run. There's sort of a base fee to cover the time on the equipment and what goes into getting it ready for each run. And it can vary from experiment to experiment, but typically each run can accommodate in the range of like 25 to 30 samples is as a very general rule of thumb. But again, this can be fairly project specific. So these are the type of things we can work out and get into some more specific details when we have a meeting about the new project. Okay. And then moving on to the third phase, where paired with each of these different groupings of acquisition, we have either targeted, untargeted, or targeted, we have a specific analysis pipeline to go with them. And so for the untargeted analysis, we have a software called Compound Discover, which is licensed through Thermo, which is basically able to take each of what I've referred to as features. So basically any ion mass charge value coupled to a specific chromatography specific retention time that have been detected across all your samples. And then it basically combines this all together, deconvolutes it, and generates abundances for each of these pairs, and then tries to identify what they actually are as much as can be. And so then this identification really comes with sort of different levels of confidence, where fall into two general categories, where one, they can be made by matching to in-house generated databases that we've derived on our own system with matching conditions to which the samples are run, which then come with the highest confidence. But then we can also probe external databases that are curated by Thermo and other labs that may not be generated under the exact same conditions as ours, but similar ones. And then we can make IDs based of other people's information too, but those then come with a lower confidence. And so just to give a little more information on how we generate these in-house databases that, like I said, are so critical for being able to ID compounds from untargeted analysis with high confidence, is basically we have hundreds of compound large library of standards that we run through our system using the exact same conditions that we will be running people's samples with. And we collect individual compound data of each one that goes into making identification. So mass and charge values of each ion. And then again, the column retention time, or basically how long it takes for the gradient to pull it off the column and push it into the mass spec for detection. And then, so basically we end up with these different, and then also like I had mentioned before, oftentimes what's called a fragmentation pattern, which I'll get into it a bit later. But basically then we build this database where for each of the compounds standards that we've run on through our system, we have these different values that go into IDing them. And then we can then sort of query that with actual sample data to make matches based on the data acquired from the samples to what we've acquired and put in this compound database. And so there's a couple different ways these IDs are then actually made, just to give you a little more sense of it. And so one is identifying them by a combination of a high accuracy mass charge measurement with the chromatography specific retention time. And so again, we're running a sample through the system using the same methods we use to generate our compound library database. And then we come out with just, a big pile or spread of different, of different mass charge values associated with different column retention times. And then basically we're just going into our, again, the same information of known compounds run through the system previously under these conditions and making identifications and assigning compounds names based on that. Alternatively, or as a complement to this, we can then also, from sample measurements, from sample measurements, also make identification by a matching to what are called fragmentation patterns or MS2 data that some of you may be familiar with. And so here, basically, we're measuring, first getting a high accuracy measurement of each intact ion, but then also after making that measurement of what becomes called the parent compound, the compound is then fragmented with specific energies, and then it breaks down into smaller mass charges. And then basically we end up with what's shown as here, although it's fairly dim, is basically what would be a fragmentation pattern. And then as an additional identification metric, we can then match how compounds in our database that were fragmented under these same conditions now compare to fragmentation patterns generated from ions detected from your actual samples. And so this was just how we make what we would call our highest confidence identifications. And then I mentioned we also can match to external databases. This is generally done with a similar process, except that they come with lower confidence ratings simply because, like I said, they are not necessarily generated under the exact same conditions as we are then running samples. But then also oftentimes there's less information that is going into the identification. So for example, you may get an identification just from an exact mass match to some compound in ChemSpider database, for example, or to some spectral library that some other lab had generated, but then we don't have our like column retention time values, which are specific to our system, to like back up that ID. And so we can, the point of this is just to get at, there are various ways we can identify, hundreds or more compounds to thousands of compounds, but that there's going to be like a tier of how confident you can be in what this ion, where it maybe is different between your samples, how well do we really know that that is what it is, just because of the nature of doing this sort of untargeted acquisition versus a more targeted approach, which people may be more familiar with, where you're oftentimes running a standard of the 10 compounds you want to measure in the same run. So you always have that information. So this is sort of a mix of trying to be able to acquire much more data, but then also be able to actually get something useful out of it at the same time. And then, so then really what you'll ultimately get out of this in terms of the deliverables from the untargeted analysis is we have planned on two different flavors of this. First is all the compounds we can ID with our, in our high confidence grouping, basically anything. So this can be, depending on the complexity of the sample, 100 to a couple hundred. And then basically for all those, we give you a list of all those with their actual abundances across your different samples. And then just some very basic statistical analyses. Although at this point, we don't plan to have the capabilities to go too heavy into the computational side of things beyond just the processing of the raw data itself that I've been talking about. But then we also want to give a second form of deliverable where we basically give you where we run our own statistics and then filter for anything that is different between your different experimental groups or samples of interest, independent of how well we can actually ID it. So here you'll get a list of anything that you might be interested in following up on, even though it may have, it may be positively identified, or it may be tentatively identified, or it may be something that is unannotated, and you just have some predicted formula, let's say, that doesn't match anything. And so this maybe is completely useless. Or for some people are interested in things that this can be a list of, well, these are things that are really different between our samples. you have, based on your sort of just initial pass identification, we don't really know what it is, but this is something maybe we want to figure out ways to pursue actually figuring out what it is. And so in a lot of cases that isn't a road people go down, because then it can become quite intensive, but we just want to give this, we don't want to just give you only the things we can ID and then throw out potentially interesting differences that some people may be worthwhile trying to follow up on. And then sort of as a less complicated process, if you are doing a more targeted analysis, we have a slightly separate analysis pipeline where for the target analysis, basically where you're generally working off of a small group or maybe 1, 2, 3, 10, whatever compounds that you're specifically interested in. And so here we're generally, like I said, unless we've run a compound before, a standard of those compounds before, under the same conditions that we're running your samples, we will oftentimes require running standards within that run to really get good positive IDs of that. And then so for this, we're talking about a much smaller list. So then we just give you abundance values and specifics, statistics for your 10 compounds of interest spanning a pathway, or whatever that might be. And so that is a more simple turnout, but then come and sort of gets more hand curation on our part, because we can easily really look at and positively identify a group of 5 to 10 compounds. But then obviously it's a much smaller list of information that you're getting out of it. But we just sort of wanted to have 2 different sort of analysis outputs that pair well with what you're actually after in your data, in the actual data analysis and acquisition end of things. And so that I'll just then leave with, so in terms of contacting, I've hit it up here a few times, but the best way at this point is just to email me, and then we can set up a meeting. Or if you have very simple questions that maybe it's not worth having a meeting of initially, I can obviously answer those, but that's the best way to reach out to me. And then there's about project inquiries, and then there's also the couple web access is kind of cut off here at the bottom, but basically if you keyword search Wilmot Shared Resources or URMC CART, these will take you to websites where us and other shared resources for the Institute are present and can take you to websites, etc. And so this is just so everyone knows the upcoming seminars. So up next, there's genomics on June 11th, and then some filled in, some not. And then I'm supposed to mention then as you may know, there's chances to win a $50 gift card if you actually register, attend, and then complete a follow-up survey. Then you'll be entered into a drawing during the August seminar. And then last, we just want to thank Dr. Vivian Pelladoro, who basically provided the funding as a donor for this to support us to be able to do these different seminars as part of the Lunch and Learn series, to basically make sure everyone knows what we can do, or people who aren't familiar with us become more familiar with us. And with that, I can just stop and take any questions that anyone might have.
So your metabolic test in mouse is for the human source of the metabolic or other species. For example, bacteria, and identify it.
So you're saying, can we do this sort of analysis of like a bacterial culture, or?
Yes, for the metabolic, they can maybe come from the human, the sample. Yes, for the human sample, maybe it also contain.
So you're saying, can we distinguish metabolites endogenous to like the human cells versus bacteria that might be infiltrated? Is that what you're asking?
Right.
Yes, I mean, to some extent, yes. So I mean, there are curated lists of, metabolites that are identified as known being endogenous to eukaryotes and shouldn't be present in bacteria or vice versa. So we can, that's not, I wouldn't say that's not part of our like base workflow, but if that's relevant to a project, then we can help with distinguishing what those are. Because we do have access, to different metabolite lists that are, what are typically called endogenous metabolites. Those would be human, mammalian, whatever, versus non-endogenous, which are things which tend to be less annotated in terms of what they actually are, but at least that are things known to generally not be present endogenously in human or mouse, whatever material.
Thank you. Could you give us an example of what any advanced sample processing would be required for like a frozen serum sample that you might have collected historically, but wanted to do some metabolomics on now?
So for, plasma or any biofluid, it's a fairly simple process where you're just thawing and then you're extracting to some extent, but you're mostly just diluting it into an organic solvent, depending on what you're actually after. And then it's just a process of cleaning up, evaporating sample typically, and then resuspending in a smaller volume of an appropriate solvent for injection into the instrument.
So, but like standard sample processing for human blood, it doesn't affect the application at all.
So you're saying like, if you're going from whole blood to plasma? Yes, so that is something that there's definitely best practices if you're processing it to do metabolomics analysis. And it's like, we have protocols we can give you that like tell you what that is. Like that upstream of processing isn't something, at least at this point, we would be doing, and don't necessarily have a ton of hands-on experience, but like we know what you should be doing to make them most applicable. So the biggest things are for plasma or serum, and it's for most things is you want to avoid blood cell rupture or contamination. And then it's basically about being more conscientious about how quickly metabolite profiles can change when taken out of their native environment. So it's about keeping things as cold as possible and doing whatever you're doing equally to each sample, because if one sample's at room temperature 10 minutes longer than another, for some things that might not matter. For this, you could have big changes going on depending what you're looking at. So it's sort of those sort of best practices that we can give specific protocols that, that people we've worked with or other cores that we're in contact with validated and have used repeatedly. Did you?
Well, what percent of the metabolites in the cell do you think you identify? Thousands, tens of thousands.
Yeah, in terms of like the small molecules, it's a very tiny fraction that you ultimately can access, where but I would say of like, in terms of what we would typically think about with metabolites, like so we can get, on the order, and it depends on the collected sample, but on the order of like around 100 to like a few hundred of like well annotated and highly confident identification. We can identify like thousands of features, so it's more about how confidently we can identify what those are, if that makes sense. And then for lipidomics, which I should have mentioned, so we've been mostly focused on the metabolomics or hydrophilic end of things. And so at this point, we're more limited in what we can do on the lipidomics end, but that with all the different, the diversity and similarities of lipid species there, we're easily measuring in the thousands of just like a typical sample, and maybe more depending on how complex it is. But that's just to give you, a rough idea. It's also very sample dependent. So like a serum or a media sample versus a chunk of small intestine, for example. And then also, so it depends on what's in there and how concentrated it is, whether we're actually at detectable levels. Cause you know what I mean? So with these untargeted, we're trying to do more a more generalist approach. We're trying to like cast as wide a net to touch on different areas of metabolism, but not necessarily get every metabolite in every pathway. And so in that sense, things that are of lower abundance that we might be able to detect if we had a specific method optimized for that, we may miss that, but we may pick up, you know what I mean, 3 of the 8 in that pathway to give you a sense if anything's going on in that pathway, but we wouldn't give like necessarily comprehensive of every metabolite of every pathway, just because like I said, it's you can't really optimize the chemistry is so diverse. It's you know, I mean, it's impossible, at least within 1 or 2 methods to optimize for everything. In the way you might be able to better capture, you know, I mean, other areas like of genome or gene
And
gene expression.
If we had a leukemia sample, how many cells would you recommend?
So you're talking like a cell culture. So
Self-sorted.
yeah, or cell sorted. I mean, so ideally we would want like 1 million cells. Yeah, we can go lower. I mean, so it's sort of, we can work with, I mean, within reason down to what you can give us. It might just limit, you know, I mean, the lower, if you can't meet sort of the minimum, then more things might drop out in terms of not being a high enough concentration. And it may limit, for example, we typically want enough material so that we can have excess material. So if the instrument has some air in the middle of a run, we can go back and run again. Whereas if you really can only get like 200,000 cells, we can do our best, but then that may be all the materials then run type of, you know what I mean? So it's sort of, again, it's flexible, but as a good rule of thumb, if you can give 1 million cells, then we can sort of do everything the way we would like to do it in terms of sample concentration and having backup material, etc.
So how reliable is the machine to measure characteristics within in tissue between animals? Like, say if you were trying to characterize a phenotype in an animal, how reliable or how many animals would you need to see a difference or likeness?
So you're getting at like how many like replicates within an experimental group?
In vivo, like if you had animal tissue.
Yeah, I mean, so typical range would be like 3 to 6, in terms of like variability of the machine. Now, depending on how much noise is in your biological system, you may need more than that. And there's going to be certain metabolites that are just more noisy or less stable and more prone to changing while the sample's being processed. So like it is a general 3 to 6, but then it can be potentially more than that depending on your system. And if you happen to be really interested in, you know, I mean, metabolites that we know to be very sensitive or that their pools can turn over very quickly as soon as they're taken out of their sort of regulated endogenous environment. But yeah, so that's sort of, I guess, the best answer I can give as a general rule of thumb.
Might there be a circadian component to it? So like, I mean, would there be variability based on when the animal was set?
Yeah.
So if you were trying to characterize a phenotype, you might want to sack the animals at the same time of day, or would that not matter?
Yeah, I mean, I don't want to dive too much into metabolism and circadian rhythm. I don't know a ton about that, but yeah, I mean, like with anything, you would, if you know certain characteristics of your system are going to influence what you're ultimately getting out of it, then you need to take that into account. So if something is dependent on whether you're sacking them at 9 a.m. versus 9 p.m., then yes, you would want to take that into account.
Well, I'm saying if you don't know, so like.
Yeah, so if I don't, yeah, so we don't.
If we don't know, so if we like use a failure to thrive model. I think what we ran into years ago when doing metabolomics on our animals where we just, we sacrificed at a humane influence. So,
Yeah.
But back then, we saw a lot of variability
and metabolites, and so one of the things that was, was discussed was that it might be, one of the factors might be
Yeah.
the time in which we took the tissue.
Yeah, so there's a lot of things that would definitely be influenced by the timing of the tissue.
And so that might change.
Yeah, so then you could either have like a pilot and like just actually little pilots there set up to actually track like what things are moving into time dependent, and then based on that, you can decide like, how do we need to set up the real experiment to like actually capture the different time points where the oscillations are occurring, for example. And so if it's just a 24-hour oscillation, then you may be able to just make sure they're all at the same time, or maybe 12, you know what I mean? So it would depend on what the circadian oscillations are, et cetera. And I'm sort of making stuff up here without knowing the specifics of what the changes, what the oscillations in circadian influence is. But yeah, I mean, that would be something where you would potentially then need to break that down and have your N of 3 to 6 at different time points, or at least make sure it's all at the same time point. If you're just looking for consistency, but not actually trying to understand the circadian oscillations themselves.
Just trying to reduce variability.
Yeah,
Yeah.
yeah.
So, like, what are the thoughts about the timeline for having the ability to do like carbon 13 flux tracing and vital studies? Is that something that's gonna happen this year, next year?
So I mean, so that's like sort of a branch, and that could be targeted or untargeted. So I mean, I would say for the untargeted, a targeted pathway, if it's something that we can figure out a method that covers that pathway as much as possible, like we can do that
So
now.
that would be doable
Yeah,
as a custom.
within whatever the instrument is able, I mean, so it would have to be something that our chemistries and whatever that we can replicate how the actual metabolites are extracted and measured. But yeah, we can do that now. And the untargeted, we've only sort of really touched a little bit on doing untargeted labeling, where you're basically taking some source and just saying, where of all these things that you're having a hard time deconvoluting, now add in all the different carbons. So like there is workflows for that, but we haven't really battle tested those yet to see how good that is. But I mean, but it seems it just adds a step of uncertainty and convolution to your data, just talking to other cores who have used a similar analysis pathway as us. But yeah, for like, if you're just trying to do like C13 flux through glycolysis, or not, I shouldn't say flux, but stable Laban or whatever, that's something that we can work with now.
Yeah, so I heard that NIH might be requiring people to share metabolomic data, just like with genomic data.
Yeah.
That could be a rumor, not really sure. but
No, yeah, that is true.
That is true. Okay, so do you provide any assistance with that or guidance for that?
Yeah, we can. I mean, basically we would, so with every, I mean, every sample ends up with a raw or semi-raw file associated with it. And so that one form of those raw or semi-raw formulas is what you would need to upload. And then that all contains any metadata that, depending on what the specific requirements are. And I think, I mean, last I saw, there's not like stringent, like it has to be this, or they're like, well, ideally we would like this, but there's also sort of minimum requirement type thing. And so, but all these files capture like all the metadata that you would need. And so we can help you sort through that depending on which parameters you actually need to upload in addition to the data files themselves.
Is there a standard database to go to?
There's like, was Metabolomics Workbench. Yeah. So that's one. Yeah. Yeah. And so this is all new. So we're trying to like keep up with a little bit and know some of it from mine and Josh's previous work and stuff. But yeah, so that's something we can definitely help with to make sure we retain and give you all the information you need to meet those. Yes.
Thank you.
Anything else?
What's the turnaround time right now? Like, what's your,
Um,
bring you samples tomorrow?
yeah, right now it's,
Then
it's,
Friday?
I would say right now it's
What's
very
your-
variable, so,
Okay.
I mean, yeah. It'd probably depend on, that's something that we could figure out like once we actually talked about the specific project. And it would also depend on how long of a line there was at the time. And so, I mean, right now it's fairly up and down, so sometimes we have like a month worth of runs already set up, and then sometimes we're just doing, we have a lull and we're working on developing our pipelines that we're sort of just at the point of putting the finishing touches on. But then that's something that we can then just work in projects into, I guess is the best way to answer that right now.
Thanks, Fred.